Here's a Renovo catheter itself. If you've never seen it before, it's kind of straightforward. You could feel free to mag in on this if you want to get a better look at this here. So let me just kind of explain to you what all these different ports are. So these are labeled here. This is the distal balloon here, which controls the balloon at the very end of the catheter here. There's going to be two balloons here. There's a proximal balloon here and a distal balloon. And if you don't ever forget, it's labeled very clearly here. Here's a distal balloon to blow up the distal balloon. This is the more proximal balloon to blow up the proximal balloon. We're not gonna adjust this right now, but later on once we have this on the wire, we could take off this little pin and to just change the distance between the balloons. Distance between the balloons goes between 15 millimeters and I believe about 109 or 110 millimeters. Typically for most of these cases, we're going usually to the minimum distance, especially in this case where we're treating the superior mesenteric artery with our branches. We really want to minimize the distance between the balloons because we want to have a stagnant column of contrast between the two balloons. If there's any branches, all the chemotherapy is going to go into the branches, and that's something that we don't want. There's two other lumens here. This is a flush port, and this is the red one is the chemo infusion port. So we're going to flush these in a second, but this is through which we're going to infuse the chemo once we have a stagnant contrast column. And this initially we just utilized it to flush it, but now what was shown in a couple animal studies, and now we're looking at it in human, is that you could actually put in our arterial line to this port, to the flush port, and you could do real-time intra-arterial pressures, and we might be able to correlate that with having a good therapeutic effect. In some of the earlier studies that have been done, if you see an increase in about 80 millimeters mercury from baseline to after you inflate the balloons and you're injecting the chemo, that may be correlated with an improved response and preventing washout of chemotherapy. So again, we might do that during this case. It's still being studied right now. So that's the catheter itself. It's in terms of the catheter, it's 014 compatible. Another really important thing, and this is something can we kind of learn from doing these procedures earlier on, is what's the best? How do we introduce this? And there's a lot of different devices you could utilize, a lot of different sheaths. My preference is a steerable sheath. It's 6 French sheath compatible. We're utilizing this Bayless sheath, and if you've never utilized this before, they come in a couple different sizes. This happens to be a 6.5 French. But basically, I'm going to put this down. As I turn it here, you can see you could angle this, in whatever direction you want. And do I need this for every single case? No, but I find it to be extremely valuable in cases like this, as it gives you a lot of stability for the catheter. You could utilize your standard Trumo destination sheath or an Ansel. There's a lot of sheaths you can use, but especially a very angulated anatomy, I think these are invaluable. If you're going to utilize this, I recommend using either a 45 or a 55 centimeter steerable sheath. The 45 I think works the best. You can use a 55 as well, although you get kind of at the end of the distance, but it still does work. So that's just a couple of things with the device itself. We're going to go ahead and flush this. You have a smaller cc, maybe a 10 cc syringe. But in terms of flushing it, I want a 3-way stopcock and a 3-way stopcock and a flow switch, please. Hey, and look at the 3-way music.
You got a 3-way here and the flow switch.
Flow switch, okay. I'm going to take a flow switch. I'm going to put it on this is the flush port here. I'm going to put a 3-way on here. This eventually will be for the chemo. And just straightforward in terms of flushing. There's nothing really special about it. It's probably going to be hard to see here, but flushing, there's some saline coming out. I'm going to lock this in place. This is going to be the chemo lumen. Again, we're going to flush this. And you're flushing between the two balloons here. Okay, I'm going to lock that in place. And then the final thing we want to do is we want to test the balloons. We typically want to use, can I get a 1 cc medallion? We put about 1/3 contrast and 2/3 saline. To basically use a 1 cc syringe to test each one of these balloons. Again, typically about 1/3 contrast, 2/3 saline. And it requires very little volume to actually inflate these balloons. You generally want to, it's pretty remarkable how little volume, how much you could go up on this. The maximum you want to put in these is about 1 cc's. I think it's really 0.99 cc's. And that gets you to a little bit over 1 centimeters. But when you're actually inflating it, in the patient, I recommend going maybe about 0.2 cc's at a time because it inflates much faster than you would think. So we're just going to test these really fast. So we're going to start here with the distal balloon. And actually, I'm going to just prep the balloon a little bit.
ACT is 242.
240. Can we give it 2,000 a heparin, please? Yeah, but before we actually put the device in, we want to make sure the patient's fully anticoagulated. I like the ACTs to be well over 250, especially in a patient that we're treating the superior mesenteric artery where we don't really have a lot of room for error. In terms of, you want to just hold this up, Kev? So here, I'm going to be blowing up the distal balloon here. I don't know if, hold it very still, and I don't know if you can mag up on here, but I'm going to blowing up here. And you see so all we're doing, we're just testing for any leaks or anything. The balloon itself, and it's hard to, unless you're feeling, but it's very soft and atraumatic, which is great for vessels like the SMA or hepatic, wherever we're treating. I've yet to see, an actual vessel injury from one of these. Another important thing, which I forgot to mention before, is that if the patient has any biliary stent or prior Whipple or any type of violation of biliary sphincter, these patients do need to be treated with antibiotics for the procedure so you don't get an infection. Everybody kind of has maybe a little different way they do this. I kind of have a rhythm, having done a decent number of these cases now. So we've already done our SMA angiogram. We're going to go ahead and put it in our steerable sheath. Next thing we're going to do is go in and select the SMA. After we select the SMA, we'll do a limited run, and then we'll place a microcatheter and a guide wire distal into the SMA. I prefer a soft guide wire at that point in time. Once I'm distal and I'm confirmed I'm in a good spot, we're going to put in a stiffer guide wire, which our stiffest 014 wire happens to be a Grand Slam, but it could be, any stiff 014 wire is my preference. And only at that point we will remove the Cobra catheter, we'll remove the microcatheter, and at that point we will advance the Renovo catheter in place. And then we'll do a bunch of projections, a bunch of runs to try to find that sweet spot in the, I'm not showing you the angiogram here, but his tumor happens to be surrounding the superior mesenteric artery. So that is our target. And that's how you determine, in terms of, when you're treating these patients, how do you determine which vessel do you treat? It's really looking at the cross-sectional imaging. The tumor surrounding the common hepatic, that's going to be the artery that we treat. If it's surrounding both the SMA and the common hepatic, maybe you're going to rotate between your treatment. A patient who had a pancreatic tail tumor, where we just treated the splenic artery. So it's really dependent on the cross-sectional anatomy to determine which vessel you're actually going to treat during each one of these interventions. I know from having treated this patient now 4 times before, it's a very challenging anatomy. The shortest distance you can go between the balloons here is 15 millimeters, and there's a little branch there, which is always a problem. So in this case, you have to pull it back very, very proximally in this situation in order to achieve that stagnant column of contrast. Next thing we're going to do is we're going to put in a stiff wire, and we're going to go in with our, like now I think we have a 5-franc sheath in place. We just give a little bit more heparin. I'd like to check another ACT in 10 minutes, making sure that we're well above 250 before we actually put the Renovo catheter in and before we inflate the balloons, because, again, we're going to be typically inflating the balloons for about 20 minutes. So especially in a territory such as the SMA, I want to make sure that we're fully anticoagulated so we don't have a complication. I've yet to have any issues with thrombosis, and I don't want this to be the first, so I think that's really critical. But in the meantime, we could, we're going to go ahead and get our steerable sheath in place. So we're going to, and by that time, it'll probably be time to check the ACT. With the amplets. The only thing that we've done, prior to this is, we have right femoral access, and we've done an SMA angiogram here. Again, this is a patient who's had already 4 treatments. You could, hold on for one second. If you just look at the angiogram here, you can see their SMA is widely patent. There's no irregularity. Everything looks good. And there's little motion here, but you could see here on the delayed images here that the SMV there, the portal vein, is patent as well. So we're in good shape. One of the things, at least in terms of the SMA injection, is if there is any SMV thrombosis is at least considered a potential contraindication to doing the procedure. So this is the steerable sheath. If you've never utilized a steerable sheath, they're really, really handy. So we just put it in here. We still have a guide wire way above that. We're taking out the dilator here. We're going to hook up this to just a flush line. Okay, so we have a continuous flush line on that. And then basically what we're going to do is I'm going to mag up a little bit just to show you better. So I'm going to pull the wire back here. We're going to go ahead and turn this. And then I know that it comes off right about there. So there you see, it just kind of popped into place. We're going to just double check that we're in the right place here. Contrast this. Can I get a wet towel? Good. So we're nicely situated in the SMA here. So I'm going to pull this microcatheter off of here. And then the next thing we have to do is pull out this Cobra so we could introduce the RenovoCatheter directly into the sheath. So wired. Good. And now we could take the RenovoCatheter. All right, so we have we have our steerable sheath in place. We have our stiff 014 wire, within the SMA distal branching, and now we're going to introduce the RenovoCatheter. So let me kind of show you the RenovoCatheter. So again, as this clip is still on here, we do not want to remove this clip until we're on the wire. We already flushed the chemo port, which is red. We flushed the flush port, and we tested the two balloons, which are fully deflated at this point. The next thing we want to do is we're going to remove this, and we're going to load this on the 014 wire.
ATP 275.
Perfect, thank you. Well, good ACT level now that we feel comfortable with. Give me 1 more 1,000 of heparin. I tend to be a little, more aggressive when we're treating the SMA. I just want to be... Bleeding I can deal with. Thrombosis is a problem. Which we've never ever had before. Do me a favor, flush the back end. I realized that we hadn't flushed it. That was my fault. We never flushed it after. There we go. Thank you. There we go. Yes. No, we're going to do that right now. So what we're going to do is, all right, so now that we're loaded on the catheter itself, on the wire itself, what we're doing now we're going to adjust the distance between the balloons. The balloon is here and here. I know it's hard to tell, but we're at the maximum distance, which is about 110. Now I'm going to pull this off, and this I'm going to pull toward me. And as I'm pulling towards me, you can see we minimize the distance. And then the minimum distance between the two balloons is 15 millimeters. So we are at the minimum distance between the two balloons right now. That's where we want to be. We are at the appropriate ACT level that I'm happy with. We've already loaded our injector with our chemo. So next thing we're going to do is we're going to introduce this catheter into the patient. Now, just to looking back at our SMA angiogram here, let's go back. You can see that there's a branch that comes off very proximally. And so we're going to basically have to put this catheter at the very, very proximal aspect of the SMA. And we're going to actually have to pull back our guiding sheath such that the proximal balloon is going to be, partially in the aorta and partially in the SMA in order to achieve a static blood flow that we need before we could introduce the chemotherapy.